To begin with, we need to talk about early development. Somites are sections of pluripotent cells which form periodically during pre-somatic mesoderm in early development of most multicellular organisms. Vertebrate embryo will segment into regular spaced blocks of tissue at periodic intervals in time in a process called somatogenesis. This periodicity is regulated by a molecular oscillator called a segmentation clock. But first we need to talk about cell signaling. Cell signaling is the communication between cells which controls all basic cell actions. The main cell signaling pathway looked at within this study was the Notch pathway. The Notch pathway is a cell signaling system which can be seen in almost all multicellular organisms that regulates cell differentiation during development, maintains the homeostasis of adult tissues, whilst mediating juxtacrine cell signaling between close contact cells. Research has shown that problems occurring in the Notch pathway can be heavily linked to the increase in disease. Mutations of Notch receptor activation can lead to adult T-cell lymphoblastic leukemia and lymphoma, whereas loss-of-function Notch receptor and ligand mutations can be found in several disorders such as Alagille syndrome. RBP-JK acts as a transcriptional repressor, meaning the expression of the targeted genes cannot occur when no ligand is bound to the extracellular domain. However, when two cells come together, a signal cascade can begin. During juxtacrine signaling between cells, a delta ligand will bind to the extracellular domain of the Notch receptor, activating the Notch pathway. This binding then cleaves the extracellular domain of the protein from the rest of the protein, leading to the cleavage of the intracellular domain by gamma secretase. This releases the Notch intracellular domain, allowing it to move to the nucleus of the cell. Once the Notch intracellular domain moves into the nucleus, the NICD binds to the transcriptional repressor RPPJK, leading to its removal. The binding of the Notch intracellular domain also promotes the recruitment of transcription factors, activating gene expression.
I'm Lucas. I'm a PhD student at KM's lab, and I'm studying the segmentation clock using Notch. Notch is really important because actually it's involved in a lot of different processes, and it's involved in how cells can communicate to each other to form the embryo properly, but also it's involved in a lot of different cancers. So it's regulated by the cell cycle, and actually it's really important on regulating this process. Because Notch is involved in the segmentation clock, we don't really know exactly how Notch can affect the pace of the segmentation clock. We have some ideas, but we don't really understand it yet.
So mathematical modelling is quite important in this paper. What is it that the maths is trying to show?
So we're trying to show that if you affect NICD half-life, you can affect the pace of the segmentation clock. So traditionally, the way that was conceived, the oscillation is just they're only regulated by Hess inhibiting itself. So most of the work that have been done in this area has been done basically on negative regulators of the segmentation clock. But what is really interesting of this paper is actually positive regulators can also affect the segmentation clock in a significant way.
A study by Kim Dale and Philip Murray looked into the idea of how intracellular oscillations are coordinated in the form of waves of expression across the presomatic mesoderm. So when asked about the significance of her discovery, Kim replied that Notch and delta oscillate at both mRNA and protein level. The first indications of dynamic expression at the level of obligate signaling components, but not just intracellular components. So this allows understanding of how neighbours can couple and synchronize. Both Notch1 and delta1 mRNA expression oscillates in the presomatic mesoderm via a clock gene, and their transcription is likely to be regulated by the segmentation clock. Kim told us that the most challenging component of studying the Notch pathway is that this is a dynamic spatiotemporal process that functions on a fast timescale where gene expression sweeps across a tissue with a periodicity that matches somite formation. It can be considered as a clock that works in an unsegmented tissue. If you think about it as a loaf of bread that is being sliced, the clock regulates how fast the slices are cut and how big they are. It was clear to us that this study had major scientific implications. Kim described these to be that understanding how Notch regulates somatogenesis had major important medical implications, as we know very little about congenital vertebral malformations that affect 1 in 1,000 people. One form of this is spondylocostal diastosis, which gives rise to malformations in the segmentation of the presomatic mesoderm, leading to abnormalities in the spine such as curvature. The Notch pathway is one of the most important and integral pathways within our body, and any future research into this area has real potential to change people's lives.